Hi there, nice to meet you. This is Heesuk Lee from Department of Science and Technology in Chung-Ang University. Okay, first of all, I really appreciate giving the opportunity for this international symposium. On today, I would like to share the information of my research with respect to the development of obesity through endocrine disrupting potentials containing the estrogen receptor agonist effects or intentionally added substances such as pesticide products and veterinary drugs in food metrics. And I'm gonna say for my presentation according to this content. Firstly, I would like to introduce my other background for my research, and I think the material, method, and result can be explained nextly. And then I would like to finish my presentation with future plans for my research. Regarding the background for my researches, oh, before starting the story of my research, I would like to introduce myself. As I mentioned, my name is Heesuk Lee, and I have researched in Department of Science and Technology, Chung-Ang University. I think my research fields can be summarized with four keywords. First keyword is risk assessment of chemical risk factors. Second keyword is endocrine disruptor. And third keyword is natural toxins containing mycotoxin, phytotoxin, and marine biotoxins. And last keyword is that the animal alternative testing method. Regarding my research interest, I think my research interest can be categorized onto three things. First categorized is that the AOP, adverse outcome pathway approaches in endocrine disrupting chemicals, that exposure of endocrine disrupting chemical and development of obesity and diabetes. And I also tried to develop the animal alternative testing method to detect these adverse effects. And second is that the risk assessment of chemical risk factors in human sample, such as urine and serum and food metrics. Also, I have tried to conduct a safety assessment of materials for food and medicines. And last is that the characterization of a natural product produced by microorganisms and plants for human hormonal health. Regarding my activities in my field, actually I have participated the OECD expert group related to endocrine disrupting chemicals. I have participated as representative Korean expert in acid validation management group of non-animal testing since 2012, and also I have participated in acid endocrine disrupt testing and assessment expert group since 2017. Also, I have joined as peer reviewer to develop the OST test guideline. And also, I have participated as the organizing community and scientific panels related to the academic society in Korea. Okay, from now on, I would like to start introduction of my research with a background. As we know, the endocrine disrupting chemicals are exogenous chemicals that interfere with the normal functions of the endocrine system, such as synthesis, secretion, transport, and metabolism of endogenous hormones. And additionally, the malfunctions in the human endocrine system can be caused by a number of products that contain endocrine disrupting chemicals such as food, plastic products, and cosmetics. So, because these chemicals can affect the endocrine systems related to estrogen, androgen, and side hormones. Actually, a number of papers published that several chemicals contained in various pesticide products have been identified as endocrine disrupting chemicals, and their adverse effects in humans are consumed as their residue persist in the environment containing the atmosphere, soil, and water, and food matrix. Furthermore, actually, a number of papers published that various previous reporters have revealed that exposure to endocrine disrupting chemical may cause a number of diseases, including breast cancer, cervical cancer, and diabetes and obesity. And to detect the endocrine disrupting potentials of a number of the chemicals to which humans can be exposed to through food, environment, and the other products, the Organization for Economic Cooperation and Development, OECD, has established the expert group in field of endocrine disrupting chemicals containing the EDTA-AZ and VMG-ECHO and VMG-NA. And the OECD has developed a test guideline since the 1990. And the OECD provided a conceptual framework for the endocrine disrupting chemicals testing and assessment, which comprised 5 distinct levels. And among these 5 levels, the level 2 outlines the in vitro data collection protocols used to ascertain the endocrine processes affected by these compounds. These test guidelines in Level 2 have been developed and validated in expert group for validation and measurement non-animal testing method. And this features is one of things in the annual meeting of OECD Fringe NA. Actually, many representative expert in each country have participated in annual meeting with OECD Business and Industry Advisory Committee (BIAC). Actually, around 20 testing methods to detect the endocrine disrupting chemicals were adopted as OECD test guideline, and the 4 detecting systems include level 2. The test guideline number 493 was adapted as OECD test guideline in level 2 firstly. And these systems can determine their binding affinity to ER alpha of their substances. And then the estrogen receptor agonistic and antagonistics can be determined by test guideline number 455. Actually, the test guideline 455 organized with 3 transcriptional activation assays, and the test guideline 455 revised in the 2 years ago. And then regarding the androgen receptor mediated endocrine disrupting chemicals, also the androgen receptor agonist and antagonist effects of substances can be determined using by test guideline 458. And actually, also test guideline 458 is organized with 3 each transcription activation assay. Actually, my developed testing method involved in test guideline 458 2020, firstly in Korea. And the last in vitro assay in level 2 is the stereogenesis assay. So actually, I applied in this study the two testing systems in these studies. The first is that the test guideline 493 estrogen receptor binding assay, and I also determined the estrogen receptor agonist effects of the fat side product and battery drug using by two test guideline for five method. Regarding the pathway related to the nuclear receptor mediated endocrine disrupting potentials, actually, OECD described that disruption of the endocrine system may occur through a number of different mechanisms, including interference with. The first is that the endogenous hormones action mediated via nuclear receptors. And second is that the hormone synthesis via sterogen or other enzymes. The third is that the metabolic activation or deactivation of hormones. The first is that the distribution of hormones to target tissues, and last is that clearance of hormones from their bodies. And actually, in particular, the endocrine disrupting chemicals can bind to various hormone receptors, such as estrogen receptor, androgen receptor, and thyroid hormone receptor, and then mimic, block, or inhibit the actions of natural hormones in this general pathway. So, a variety of environmental chemicals may act similarly to sexual steroids by altering nuclear receptor signaling and gene transactivations. And so when ligands bind to the nuclear receptors within cells, the ligand receptor complex undergoes conformational changes that leads to receptor activation and transcriptional responses. So, therefore, the transcriptional activation mediated by xenobiotic chemicals is a plausible and accepted starting point for evaluating their potential endocrine hazard of chemicals. And then I extract the water clot with cures of the endocrine disrupting chemical and obesity. So this water cloud is showing the relations between endocrine disrupting chemicals and obesity. Actually, as we know recently, various epidemiological studies have reported that exposure to endocrine disrupting chemicals leads to obesity by interfering with adipocyte differentiation and or lipid metabolism. For example, the environmental estrogenic chemicals containing the bisphenols, parabens, phthalates, and the nonylphenols have been shown to promote adipocyte differentiation and or proliferation and increasing their body weight. And regarding the material and method for my researches, As I mentioned, I applied to transcriptional activation in vitro assays to determine the estrogen receptor agonist effects of tested pesticide product and veterinary drug. First transcriptional activation assay is HERA 9903 estrogen receptor transcriptional activation assay using a stably transformed estrogen-responsive human cervical tumor cell line, HERA, with birth human estrogen receptor alpha expression vector and a firefly respirator reporter vector bearing estrogen responsive element. Actually, the estrogen receptor agonist testing method was adopted as OECD test guideline number 455 in 2009. And second transcriptional activation in vitro assay is VM7 estrogen receptor transcriptional activation assay using a stably transformed estrogen responsive human breast tumor cell line with a firefly respirator reporter vector bearing estrogen responsive element. Also, the estrogen receptor agonist testing method was adopted as OECD test guideline number 455 in 2012. And as we know, to elucidate the molecular mechanism of action of endocrine disrupting chemicals in adipocyte differentiation and lipid metabolism, 3T3L1 cell line is a well-characterized cell line often used. 3T3L1 cell line model is widely used to study adipocyte differentiation in vitro, where fibroblast-like free adipocytes are differentiated into mature and lipid-filled adipocytes through a multi-stage process. Additionally, estrogen receptor alpha also expressed in 3T3L1 adipocyte. Due to that, I confirmed the inducing the adipogenesis by pesticide product and veterinary drug based on their estrogen receptor agonist effects in 3T3L1 adipocyte according to these differentiation conditions. From now on, I would like to introduce the results from my researches. And the first result from my research is that regarding the binding affinities to estrogen receptor alpha, the diagenome FOXIM, terpenes, and tolcurpers methyl have binding affinities to estrogen receptor alpha with its 50 values like this figure. And the respiratory signal increased by adding diagenome FOXIM, terpenes, and tolcurpers methyl in a dose-dependent manner with their P10 values like figures. We are sure that it seems that the estrogen receptor alpha binding affinities of 4 pesticide products induced their transcriptional activation. And to confirm whether lipid droplet accumulation induced by for peptide product involves estrogen receptor. Repeat droplet accumulation was measured in the absence and presence of the endocrine receptor antagonist ICI-18278O. The result showed that intercellular lipid accumulation induced by for peptide product was significantly inhibited by ER antagonist. And to investigate the potential mechanism of lipogenesis by for peptide product, the expression level of proteins related to adipogenesis was quantified, and treatment with for peptide product significantly increased the expression of PPAR gamma, CDAS, EBP alpha, and peripin, and their expression was suppressed by ER selective antagonist, like this figure. And in case of representative beta-renal drug altinogest, it showed the estrogen receptor agonist effect in OECD test guideline number 455. And we could confirm this luciferase signal is based on selective binding affinity to ER alpha. The estrogen receptor agonistic signaling curve was blotted ER estrogen receptor alpha selective inhibitor MPP. On the other hand, luciferase signal induced by altinogest was not suppressed in presence of the estrogen receptor beta selective inhibitor PHTPP. To confirm whether lipid droplet accumulation induced by altinogest involves estrogen receptor alpha, lipid droplet accumulation was measured in the absence and presence of the estrogen receptor alpha selective inhibitor MPP. The result showed that intercellular lipid accumulation induced by altinogest was significantly inhibited by ER estrogen receptor alpha selective inhibitor MPP. And from the data of Western blottings, treatment with altinogest significantly increased the expression of related proteins containing PPAR gamma, CDAS, EBP alpha, and peripin, and their expressions were suppressed by ER alpha selective inhibitor MPP, like these figures. Lastly, I would like to say with respect to future plans for my researches. Firstly, I have tried to clarify the relationship between structural characterization and estrogen receptor-mediated endocrine disruptive potentials containing ER agonist effect and adipogenesis using prediction model. Second future plan is that the development of in vitro or ex vivo testing system to determine the endocrine disrupting potentials or chemicals and their metabolite. I think as we know, in vitro testing systems have a critical limitation to determine the adverse effects of chemicals after hepatic metabolism. So, I have tried to develop the in vitro or ex vivo testing system to determine the endocrine disrupting potentials of chemicals and their metabolite using S9 friction or liver organoid. And I'm sure these approaches can help avoid the hurdles in critical limitation of in vitro testing method. Okay, actually I wish we will discuss on my or your research by face-to-face meeting sooner. And if you have interesting for my research, please visit my homepage. Okay, thank you very much for your listening over my presentation. Okay, goodbye.